Hi, it's Dr. Rosario again. In our last video, I talked about how viruses are different from other microorganisms. In this video, we are going to focus on the structure of viruses. The main goal for this video is to identify the different components of viral structure. We are going to define terms like virion, capsid, and capsomere. We will talk a little more about the viral envelope and where it comes from. And we will discuss the different types of nucleic acid that we find within viruses. Some viruses may have additional components, and we will talk about that as well. Finally, we will discuss the ways that we classify viruses. The word virion refers to the completely assembled virus that is capable of establishing an infection in a host cell. So a virion is found outside of host cells. Remember, a virus is not considered alive. It does not have an internal metabolism and cannot really do anything outside the host. At a minimum, the complete virion contains nucleic acid, which can either be DNA or RNA, but it's never going to be both. Surrounding that nucleic acid is a protein coating called the capsid. Together, these form a structure that is called the nucleocapsid. Some viruses have an envelope that is composed of phospholipids. Here on the right-hand side of your screen, we have two different viruses. The one on the top is called a naked virus because it is composed only of the nucleocapsid. The one on the bottom is an enveloped virus, with the nucleocapsid surrounded by a phospholipid bilayer. Whether or not a virus has an envelope, the proteins that extend outward from the surface and are involved in binding to host cells are called spikes. You can see that the spike proteins are part of the capsid in the naked virus or are found embedded in the envelope of an enveloped virus. Viral shape is determined by the capsid. The individual proteins within the capsid are called capsomeres. There are three main types of viral shape: helical, icosahedral, and complex. Helical and icosahedral are the most common. In helical viruses, the nucleocapsid forms a long coil. This structure can form a rigid rod shape, like we see with the tobacco mosaic virus, pictured in the upper right-hand corner of your screen. On the other hand, it can be flexible, like we see with the Ebola virus, which is pictured in the lower right-hand corner of your screen. This virus folds in on itself, forming a characteristic shepherd's crook-like structure. In some viruses, the helical nucleocapsid is packaged inside of a lipid envelope. The virus at the center of this page is an example of an enveloped helical virus, vesicular stomatitis virus. In this case, the helix is folded up within a bullet-shaped outer envelope. Another common viral shape is the icosahedral capsid. An icosahedral is a 20-sided geometric shape. It is made up of 20 equilateral triangles that are formed by the capsomere proteins. The nucleic acid of these viruses is found inside this protein shell. Here we have an illustration of norovirus, a common cause of viral gastroenteritis. This virus contains RNA inside that protein capsid. Most viruses are either helical or icosahedral. Some viruses don't quite fit into either category, and we describe their structure as complex. For example, the variola virus, which causes smallpox and is pictured on the left, has this unique brick-like structure, with the viral DNA packaged in a dumbbell shape at the center of the virus. Another example of complex viruses are the bacteriophage. These are viruses that infect bacterial cells. Bacteriophage have a shape that looks a little bit like an alien spaceship. The head of the virus is composed of capsomeres that enclose the viral DNA. A helical tube-like structure connects the head to spider-like protein legs. This distinctive structure allows the virus to attach to the cell wall of bacteria and act like a syringe, injecting the viral DNA into the cell. These are just two examples of viruses that don't fit into the categories of helical or icosahedral, but most are going to fit into those categories. As I mentioned earlier, some viruses are naked, meaning their external surface is composed of the capsid proteins, while others are enveloped. An enveloped virus obtains a phospholipid coating as it buds through host membranes. As you can see in this illustration, during the viral assembly process, viral proteins, which are pictured in red, are embedded into the membrane of the host cell and then picked up along with the envelope as the virus buds outward. All of an organism's genetic information is stored within its genome. In cells, the genome is composed of double-stranded DNA. In prokaryotes, the DNA is arranged in a single circular chromosome, right? In eukaryotes, the DNA is divided up into multiple linear chromosomes. DNA is the information storage molecule. Cells access that information by making RNA, which is then in turn used to make proteins. Viruses don't always follow these same rules. Viral genomes can be composed of DNA or RNA, but you will not find both DNA and RNA within a virus. While prokaryotes or eukaryotes may have several thousand genes, a viral genome contains just a few genes. For example, the HIV genome is pictured here, and it encodes just 9 genes which are used to make 15 different proteins. Viral genomes can be linear or circular. They can be composed of just one piece of nucleic acid or segmented into multiple pieces. While the genomes of cells are composed of double-stranded DNA, viral genomes are highly variable. Some viral genomes are also composed of double-stranded DNA. Some are composed of single-stranded DNA. Others are composed of RNA, and that RNA can be double-stranded or single-stranded. If that single-stranded RNA can be immediately read and translated into proteins, we call it plus-sense RNA. However, if the cell has to convert that RNA into a form that can be read, it is called negative-sense RNA. As we'll see in our next lecture, this becomes really important when we talk about viral life cycles. In addition to the viral nucleic acid, some viruses carry additional molecules along with them. These molecules are made by the host cell and packaged inside the virus as it's assembled. Some viruses carry enzymes with them that are essential for the virus to start its replication cycle in the next host. These enzymes might be polymerases or replicases that are used to make viral nucleic acids, or they may include reverse transcriptase, which is used by HIV to convert RNA to DNA. Other things that might be packaged inside the virus may be host ribosomes or tRNA molecules. Again, these things can be used to increase the efficiency of viral replication within the next host cell. Now that we've discussed the structure of viruses, let's turn our attention to viral classification. The International Committee on the Taxonomy of Viruses is responsible for classifying and naming viruses. Traditionally, a variety of characteristics were used to place viruses in different groups or families. These characteristics included viral genetics, chemistry, shape, replication process, and the type of host that was infected. However, as we use new sequencing tools to decode viral genomes, classification has become increasingly complex. Viruses that were once considered related based on their physical characteristics have been found to have very different genome sequences and then have been classified now into different groups. For this class, we will focus on a simpler classification scheme, and this is called the Baltimore classification. This system was created in 1971 by a virologist named David Baltimore, and it divides viruses into 7 major groups based on their type of viral genome. This classification is useful because of the link between the viral genome structure and the viral life cycle, which is what we're going to talk about in our next video.